Backtracking just a bit to recap, what you see here is the slide that we looked at yesterday that showed a number of structures, and I gave some additional structures that I wanted you to take note of. So we see the three layers of the developing embryo. We see the ectoderm, the endoderm, and the mesoderm at the top here. So I'm trying to just show you these areas in here, and those areas which also can be the area that's right there in the middle, that thing we called a notochord. And the notochord is something we'd see very early on in embryonic development of all organisms, which we call chordates. Remember that a chordate is something that ultimately we're pretty familiar with because most chordates have some type of a cord, or not always a full spinal cord like ours, but we see an organism that has this midline structure, which typically then will develop into the bilateral structure that we are. So something that has a head region and a tail region and a right side and a left side, and that's what we can kind of see there on the right. We also know that there are these things called somites, and somites are some aggregations of cells in the mesoderm, and I say aggregation, I just mean there's a cluster of cells, and that the somites send out signals to determine some of the fates of some of the peripheral nervous system neurons. So when we say send out signals, we're looking at chemical signals that are produced by certain cells. Remember that from biology. Some type of a protein that would be excreted and then serve to guide, in this case, some of the cells in certain directions. So that helps to tell cells where to go, because in this early stage we have a lot of cells and they're proliferating, they're developing really rapidly, but how do they know what to do and where to go? At the very bottom figure, you see that central canal, and then you see, which is the part that is going to run all the way up and down the neural tube, and ultimately will be a part of the ventricles. Remember, the ventricles in the developed brain we see are the places where cerebral spinal fluid is created by cells called choroid plexus cells, and that it's circulated throughout the central nervous system. We saw this, you want to know these vesicle stages, sort of the early stage when we see the neural tube start to develop into these three areas, prosencephalon, or ultimately what is as a group called the forebrain, mesencephalon or midbrain, and rhombencephalon, which is the hindbrain, and then the spinal cord. And when we say vesicles, so you say, well, those are four things, aren't they? But really we're looking at the three sort of swellings of part of the top of the tube, which are ultimately then going to develop further into this 5-vesicle stage, where we see the prosencephalon further develop into these two areas, the telencephalon and the diencephalon. We know that the diencephalon is where we find all the thalamic structures, the thalamus, the hypothalamus, epithalamus. There's more thalami than you know, but that's the area. And what's also interesting is it's from this diencephalon that a structure called eye cups actually starts to develop, and that's going to connect with the retina, the peripheral nervous system retina. Then down below, mesencephalon or midbrain stays the same, and then the rhombencephalon further develops into those two areas, metencephalon, which is going to further develop into the pons and the cerebellum, and the myelencephalon, which will ultimately continue its development into the medulla and the spinal cord. And there you can see the central canal running down the middle of that neural tube. My drawing below is just to show you that prior to what you see above, to remind you that neural tube, which we saw sort of in cross-section, keep going and develop, we saw the ectoderm, some of it fold in, And then ultimately it will fuse, and it will fuse all along that midline where that purple part of the structure is, and to form an entire sealed central nervous system. So these pictures we've seen already, so I'm going to keep going. And then we're looking at now the 5 phases of neurodevelopment, the neural plate induction, which is our big focus here, a little bit on neural proliferation, in other words, the cells that make lots of copies, and those cells which then have to go somewhere. And then the going somewhere part is migration and aggregation. So migration just means to migrate, right, to move. And then to aggregate is to form clusters or groups, depending on what their purpose is going to be ultimately in the developed organism. Finally, we'll look just very briefly at axon growth and synapse formation, and then the importance of cell death and synapse rearrangement in the sort of pruning and refining of this nervous system. So a lot of this is backtracking. It's a little redundant, but you can see that we get this neural plate that develops, this part on the dorsal surface of the embryo, which is part of that the ectoderm. And we mentioned already that the notochord is something that's really important, and it actually is super important because it actually it induces the development of the neural plate. So, and it does so, and other chemical signals will also do so, to allow this development, allow neurons to do what they need to do. And often we call some of these, there's groups of these that are called growth factors. Fibroblast growth factors, or FGFs, they're a whole family of different proteins that are produced and secreted by cells and help to allow growth and development. The neural plate has these different cells, and what I want you to, well, actually the whole embryo has these different cells. So when we look at a zygote, just the beginning stages of development, those cells are what we call totipotent. That means that they have the capability of being anything. And so totipotent means that any one of those cells we could pull out, and if there was some way to stimulate that cell to continue development, it could become anything. It could become a brain, a kidney, a liver, a skin, etc. So those are totipotent stem cells. Pluripotent are ones that start to develop a little bit later, and they are also called stem cells, but these are a little less free to do any kind of development, but they still have a huge capability of developing to a lot of different types of organs. They can be differentiated. Then ultimately, some of the cells, in particular in certain areas such as the neural tube, they become multipotent, which are cells that specifically can develop into a lot of different types of nervous system cells. So they will develop into neurons, but they'll develop into some other, many types of neurons, and we'll probably not really get into the weeds on that. That's a lot to talk about, but I just wanted you to understand that in the early stages of fetal development, we see these cells which have this capability of being pretty much anything. That's the totipotent cell, and then they become pluripotent, which means that they still have huge range of capability, and then ultimately when we talk about multipotent cells, we're looking at ones in particular areas that still have a lot of capability of becoming other types of cells within that area, and that would be determined based on some of the signaling molecules that allow them to further develop. So neural proliferation is the next one. Remember, that means to really develop quickly, to grow, to do rapid mitosis. And we've seen this already. So this isn't new. It's just saying that this is actually what we're observing. We're seeing how these different areas are growing and expanding and further developing. That's neural proliferation. Migration, it means that these are going to go somewhere, right? And one of the things we've already mentioned, and you saw this in a little video I showed you, is that these baby neurons have to go to the right place, and they have to get set up in the right area of the brain. So they continue to differentiate even as they're moving. And as they do that, as they differentiate and develop, they begin to reach some potential. In other words, what they can literally do, what's their job in the nervous system when it's mature. So migration, though, has to do with the glial cells. And this group of cells, this was on this test, it was on radial glia, is a group of glia that actually act to guide these newly developing nervous system cells into the areas that they need to go. So the radial glia is that thing that you're seeing actually in B, where the neurons are actually migrating up the radial glia. So it's kind of like climbing up a pole or a ladder. It's going upward, and the radial glia are guiding that neuron that's moving into some area of the cortex, and it's moving from the radial glia operate very often from the ventricular zone. So if you see down below, it's showing that ventricular zone, the ventricle. This is where a lot of the radial glia are produced, and then they move from there up into those developing areas of the cortex. When the neuroglia, so now a little bit further along, we have all the migration has happened, the neurons are where they need to be, and then they start to mature. So they're going to develop the axons and the dendrites that they need to function properly. And there are more chemicals, as you might guess, which actually act to direct even the axons and the axon terminals toward certain areas. And these chemicals are chemoattractants. And there's actually chemorepellents too, which kind of like steer the neurons, axons and the axon terminals to go in different directions so that they go to the proper places. So that's axon growth. And sometimes that's also called synaptogenesis, which means that it's the origin, it's the birth in a sense of these structures that are these areas between communicating neurons in the brain, which we call synapses. Growth cones, you don't need to know these, but it's really impressive to see these in a video. I can show this to you if you're interested, but these are at the end, and they actually crawl forwards. You can see them moving in the nervous system as they're pulling or pushing different neurons to where they need to be. Okay, so in brief, synaptogenesis is how new synapses are formed. And these will depend a lot on astrocytes. So just knowing what synaptogenesis is sufficient, but the glial cells that we call astrocytes are important here. And then finally, cell death. And in neuronal death, this is interesting because a lot of the neurons that are made, as a matter of fact, a huge percentage of them are going to die. And they're going to die for a reason, and that's because they're overproduced, and now we need to prune it out to make these neurons that are really well set up. These are the ones we want to stay, and then all the rest of them we want to eliminate. So if they don't get to where they need to go, then there are chemical processes that go on. You might remember apoptosis, that are going to cause some of these neurons to die because they haven't been able to compete really for the chemicals to get to those targets. And those chemicals called neurotrophins are the ones that are going to help all along the way. So that's also kind of a generalized term for all these things that help the growth. And then there's going to be a little bit of synaptic rearrangement, but we're going to get to that later on. And we'll quit probably right there.